The harmful effect we can summarize into a genetic disease, genetic diseases or genetic disorders. So beneficial effect we can place into variation. And the other thing is, it's very difficult to use words like frequently, often, rarely, the frequency of this kind of a thing. We have no records. There's no statistical records to show what kind of, how often this happened, how this happened, this happened. We don't have records. The effect we need to keep, it's a balance between harmful and beneficial, because variation is the key factor which drive this natural selection process. If there's no natural selection process, there is no change of the allele frequencies. If there's no allele frequency change, no evolution. No evolution means organism cannot exist. So on top of that, everything survive on this world because of these mutations and variation bring from this variation. And this is important for whole species, not individually. At the end, it goes to the whole species. On the other hand, if someone ended up with a genetic disease due to mutation, it's more a personal thing, like affected by one person. It could go to the next generation, but it's more towards the person. But this is for the whole species. So then harmful and beneficial effect, we have to carefully balanced. We have to carefully study due to the mutations. Now, last, I try to take these wordings, the Campbell Biology and Resource Book, so I just like to highlight. So if the mutation happened during the DNA replication in the gametes, and it can pass to the next generation. And no effect. There's no effect or harmful. And they say small percentage can be beneficial. So they use this word, a small percentage. Now, I personally don't like this small percentage. If that case, they have to give what is the percentage? 2%, 1%, 5% should be given here. And here, vast majority of such changes, no effect or harmful. Anyway, if we just try to take the summary, it could be no effect, it could be harmful effect, it could be beneficial effect. Now, as I mentioned, no effect, harmful effect, beneficial effect. Now, I further mention what is beneficial, variation, harmful, genetic disease, no effect. You can learn when we learning the genetic code. Okay. In a harmful or whatever, beneficial, whatever the case, mutation are the original source of variations, which is important for the natural selections. So I would say like beneficial effects more coming with variation for natural selection. The balance between complete fidelity of DNA replication, fidelity is accuracy of DNA replication, or we have learned the repair, mismatch repair, the DNA excitation repair, all these things, make low mutation rate, long period of time, allow evolution to reach diversity of species. Now, what Campbell books suggest, these mutations are at the end important for the evolution, because all the repair mechanism, proofreading, first proofreading, second the mismatch repair process, third DNA excitation repair process, they try to keep it very low. So then considering the genetic diseases and all these things are very low, but benefit is coming for the whole population, the variations. So variation is very powerful for the whole population and existence of organism, and also to make a new organism. New species arise from existing species. So then now you see what I try to explain, and I match with the Campbell biology one. Now we're going to the resource book part, what they're going to say. Mutation or collection of mutation can make cell malignant and result in cancer. Now, the first priority is not the cancer, because cancer mutation effect of cancer is later stage process. You know, any person can understand cancer is developed by the person at their late stage. Most of the people after their 30s, a single person can develop a cancer, and cancer is not spreading as an infectious disease. It's a condition due to mutation of DNA, right? So it just gives the first priority for cancer, result in cancer. Mutation also alter the phenotype, and very often they are either lethal or at least produce unfavorable phenotype. So it says different thing. It changed the phenotype. Okay, phenotype mean uh, this harmful effect or beneficial effect. So alter phenotype mean uh, no effect and effect. The Campbell biology say no effect or effect. Effect is harmful or beneficial. So then here it only summarize this effect is often lethal. So they use this often lethal. It is I not much clear. Frequently, it is lethal. So all the genetic disorders are not lethal. People can live with the genetic disease. Many genetic disorders, they are living at least a certain stage of their life with the genetic disease, and few of the identified genetic disease, immediately after birth, the baby died, or maybe after a few years later, baby died. There are rare genetic disorders like this, they get lethal. Most of the time, they can live with difficulty in lifestyle. When the mutation occur in self-producing gametes, they may be passed into the next generation, resulting in variation among species. So again, it says variations are important. It can pass to the next generation. Now I explain you the summary of mutations, taking Campbell resource book and also my opinion. It's very difficult to make a very clear sentence, clear things when we talk about mutation. As a result, people who are making the questions, they should be very careful to precisely make the question that student can find the correct answer. If they are giving large space to think, then the informations are too much. It's very difficult to understand. So you can see, I think you as a student, you can understand, uh, especially this sentence of Campbell biology, the balance between complete fidelity of DNA replication or repair and low mutation rate has, over a long period of time, allow evolution of the rich diversity of species we see. So overall, the mutations explained in this sentence. We can't take a small example. We cannot specify everything taking one example. I think this is the overall expression of the mutation. So this sentence well explained overall expression of overall impression of the mutation. So balance between harmful effect, but still low because we have a repair process and everything. On the other hand, we need to keep this kind of mutations. We cannot stop mutations because evolution is the main factor keep the organism on the planet of Earth. Okay, with that, I complete this lesson 4. Do you have any questions?
Uh, so that means with the mutations, only the evolution variations arise and evolutions come with the natural selection process, no, sir?
Yes, because uh, variations come at different points. For example, random fertilization. And we also learn crossing over and random assortment during prophase 1 and metaphase 1. They can bring changes, but new changes. If you want to bring new alleles, mutation is the only force can bring new alleles. Other things are shuffling the cards. So if you have a card pack, it's shuffling the card pack. You have only 52 cards you are shuffling and then you divide to make to the other persons. But when you add new card to the card pack, then it's new card. The mutation is actually bringing new allele. So without new allele, we cannot keep this evolution. So that means one hand we can argue randomly we have to make a mutation. When they make random mutation, there could be bad thing happen. But bad thing is not affecting whole population. But good thing come with the variation is beneficial to the whole population. So that's why mutation is difficult to express certain times because people who wants to think about cancer, they are overloaded with they think cancer is the one of the important thing. They bring mutation to the cancer, and people think genetic diseases are the most important for them. They think okay, mutation are bad, they bring genetic disorders, but overall cancer can happen, genetic diseases can happen, but their rate is we can keep low. But on the other hand, what people can't see clearly, evolution, natural selection, because all the population in this time after billions of years they're surviving because of natural selection and evolution. But people can't see this. But people can see cancer patient, people can see a genetic disease person, but evolution is the one not they can't see. That's why it's mostly going like favorable direction, it's unfavorable. But I think on the top we should keep this Campbell biology last sentence. I think it explained well. There is a mutation, and this mutation keep them low. So as a result, we have this harmful effect. On the other hand, this is important, keep on going for evolution. So that's, I think, the overall message about the mutation. But if they want to make a question specifically to certain cases, they are really specified. For example, the person is exposed to continuously to ultraviolet light, and then what kind of a thing can happen? Then you can say there are many mutations can happen, DNA damage can happen. As a result, cannot control the cell cycle. Then cell cycle going unstoppable way. More and more cells produce. This is cancer. So we can bring questions specific to certain case. We can bring question to genetic disorders. But overall, when we are making a questions, taking mutation as the main factor, only correct concept is this one.
Uh, so the genetic diseases like color blindness and stuff, uh, how should we get them? Like,
Yeah, when it comes to genetic diseases, there are many genetic diseases, different type of genetic diseases. So certain diseases, the mutations are different. Example, one genetic disease, only single nucleotide change. Single nucleotide change. After that, one amino acid change of the protein. Single nucleotide change, one amino acid change. The protein molecule, let's say it's enzyme. So this enzyme has one amino acid change. Then this can cause a mutation. This can cause genetic disease. So example, when you take hemoglobin molecule has 1, 2, 3, 4. Four globin units. So these globin units, alpha, go to the next one, okay. So in black color, this is alpha, this is beta, and this is beta. So this make the hemoglobin molecule, and the middle there is a heme group with ferrous ion. Now we have identified one mutation at one position of this beta globin. If this is the 3 nucleotide, so one base change let's say this T change to A. Example, I'm not taking 100% accurate one. So then you see there is a one nucleotide change. Now this can cause complete change of this protein. As a result, the hemoglobin molecule, the total hemoglobin molecules, the shape become like this. It should be like this. Now it is like this now. Just only one nucleotide change, it changed completely structure of proteins. And this condition we're going to learn as a sickle cell anemia. That's a red blood cell. Let's say like this. So red blood cell get this kind of a shape instead of this biconcave shape. Just changing one nucleotide. Now this brings some kind of fear. This is very sensitive. One nucleotide change ended up in new genetic disease. But I took an example. In certain cases, there are many bases deleted. Like some cases like 40 genes deleted. And they again ended up in the genetic disease. So it's very different genetic disease and its mutation. So we have to learn case-wise. We take one genetic disease and we go to check what is the mutations. We cannot generalize mutation with all genetic disorders. This is one example. Single nucleotide change can bring genetic disorder. So if we take another different genetic diseases, their base changes are different. I think when it comes to genetic diseases, we'll take one by one, and then we learn genetic disease. And this we talk about the mutation in the genes, and we can have to talk about mutation in the chromosome level, the complete chromosome change. That is also mutations. We have to talk about that as well. We'll learn it in the coming up lessons. This lesson summary is there are three processes we can highlight: proofreading, mismatch repair, DNA excitation repair process, nucleotide
Uh,
excitation repair process. So,
so in germ cells, if it occurs, we can get genetic diseases, but if it occurs in our normal body cells only, we get cancer, right?
yeah, yeah. So we can generalize into three actually. This I get the space I will show you. In this case, mm, the parents, just get here. So we have learned about this uh, genetics. If this allele simple A is mutated one, and offspring get a chance 25% of chance getting the simple allele, then 25% of chance the person can get genetic disease. This is from parents to offspring inherited, so it can pass to the offspring. But in certain cases, we can also say the parents has completely healthy, even they don't have mutated alleles. But there are again chances the offspring have a genetic disease during gametes formation. So parents, they are okay, but gamete formation, there could be mutation happen. Then offspring, they can ended up with the genetic disease. The third situation, uh, adult stage, exposure to UV rays can get mutation, and this is the cancer. So this is ended up in a cancer. So these are the three scenarios we can highlight. If the parents have a genetic disorder, it goes to the offspring. Parents do not have a genetic disorder, but gamete formation, if genetic disorder happen, something happen, it can go to the parents. I mean, I can give a small example, not maybe not the best. For example, Down syndrome. Down syndrome is having a chromosome mutation. There is a three chromosomes come into position 16. So there are 22 autosomes. At 16 position, there's a trisomy. Three chromosomes come, and this is called Down syndrome. Parents are completely healthy and they don't have any genetic disorders, nothing. But the baby born with Down syndrome has a chromosome change. So that kind of a gametes formation, the chromosome are not properly aligned, properly distributed, so it can happen. And cancer is a very common conditions due to exposure to harmful things like UV radiation and chemicals. This is again mutation happens in the certain cells of the body, and these cells cannot control the cell cycle. For example, skin. If it is happen in the skin cells are start to divide. So come as a skin cancer. So these are the three scenario we can find in mutations, the harmful effect of mutations. So is it clear? We have done the first part of this lesson now with the fourth lesson.
Uh,
Understanding the DNA replication and significance, and its repair process, everything we learn.
Clear, sir.
Okay, according to our notes, so notes mean we use a resource book as our notes, but here and there are some mutation in this resource book. It's okay. The second one is gene and how they work, the nature of prokaryotic and eukaryotic genes. Now we're going to learn here more about genes. If you look genetics, we learn so many things, we discuss so many things, but we haven't learned about genes. And here we look DNA molecule, not the genes. So here we get a chance to look how prokaryotic and eukaryotic genes look like. And what are the important things we have to understand? What are the important concepts uh, we have to follow in prokaryotic and eukaryotic genes structure and its function? Okay. Now, so far, when we are learning about genetics, so Mendel, he didn't know about the genes, but he identified certain patterns of this gene behavior. So he used this term heredity factors. So he tried to imagine something closely related to gene, but his intelligence did not reach that level to come to the gene level. But he understood something there in these cells. They behave like this. So basically, we can call these heredity factors. There are factors, and these factors exist. This factor causes certain phenotype, and in gamete formation, this factor is going to the gametes, separate into gametes, and when two cells fuse again, these factors come into action, and one can give this character, other cannot. Like this, he just try to imagine the whole scenario of the gene behavior. So it all come under these hereditary factors. But what happened when we are learning genetics? We completely uh, bring this concept, new concept. We mentioned this is allele, this is locus, this is gene, and this is a recessive, this is a dominant. We all add this uh, new knowledge to understand the purpose. If we imagine if we learn genetics without new knowledge, we have to use, there is a factor. We can't use term allele. There is a one factor. It comes from this cell. Like that, we cannot learn genetics well without bringing the new knowledge. So we have to bring the new knowledge to learn all genetics. But if we look from the Mendel view, he doesn't know anything. He used the hereditary factors. So we come to the next lesson, how people discovered these hereditary factors to chromosome and gene, this transition period 1900 around. So they identified, okay, what Mendel said is something this. So we have to use this term, we have to use this uh, process to explain genetics. Now, we discuss so much things about the chromosomes, uh, locations, alleles, and diploid, haploid, like all we discussed. I think we can directly going into the correct point. So when it comes to a eukaryotic DNA molecules, uh, we know that chromosomes, where is it this? Okay. The chromosomes are linear, and along the chromosomes, we can find regions, and these regions are termed as genes. That's what we learn. And we also learn the chromosome exists in pairs. As a result, we call homologous chromosomes, and gene exist in pairs then. So we call them allele. So that's what we learn in diploid cells. Alleles are the gene exists in pairs. We call them allele, alternative versions. That's it. So now we're going to further explore what is molecular level, what are these things, what kind of a thing present, and why they are there, and what are the other things we have, like thing we're going to learn. When you take the prokaryotic chromosome, prokaryotic DNA, they have a circular DNA. And in this circular DNA, again, we can find genes. Okay, we're going to highlight few differences, eukaryotes and prokaryotic DNA. One thing is the eukaryotic DNA molecule is huge compared to the prokaryote DNA molecules. Prokaryote has a smaller DNA molecule and eukaryote has larger DNA molecules. As a result, there is one interesting thing we can observe. 98%, they are not responsible to give a genes. They're called non-coding. They are non-coding region. Only 2% represent the gene. Whole DNA molecule, only 2% represent the genes. 98%, they are not genes. On the other hand, prokaryotes, almost we can consider most of the portion, they are genes. So we cannot say the number wise what is exactly for prokaryotes, but they are mostly represent the genes. They don't have these non-coding like things. Most of them are genes. Another difference is the prokaryotes. If there's a gene like this, and there's another gene adjacent to that, and there's another gene adjacent to that, like this, gene 1, gene 2, gene 3, like this. So these 3 genes at the same time going to form a proteins. Protein 1, protein 2, protein 3. But when it comes to the eukaryotes, if there's a gene, that's a one gene, and it goes to make a protein. That means in eukaryotes, single gene at a time operate to make a proteins. But prokaryotes, many genes at the same times produce proteins, different proteins. We'll come to that a little bit later on. What is this process of eukaryotic and prokaryote protein production like things? Before that, we're going to learn the eukaryotes. What is this non-coding region? Right. By our original definition, we try to explain a gene is a sequence of nucleotide which can provide codes to make polypeptide chain. So this is a gene. Gene is a sequence of DNA provide codes to make polypeptide chain. After that, polypeptide chain altered to make the protein. So that is a definition of a gene. Then there's another gene at this level. Here is another gene. So in between, what we get the DNA molecules. This is the DNA molecule, right? It's going like this. I look at here, the middle, this place. So I'm going to tell that is not a gene. So what is that then? This space is called intergenic region. Simply a non-coding part. So that's doesn't make a protein, that doesn't make a polypeptide, that doesn't give a code to make a polypeptide. So its function is not to give a code to make a protein. These regions are called intergenic region. Now, intergenic region, you will learn in the future if divided into again many regions. So people try to identify function of these intergenic regions, and based on that they try to give names like science, NES science, like this kind of names given for the intergenic regions, and scientists trying to find all the functions and significance of intergenic region, because at the moment gene we know how it operate, how it copied, how it make the proteins, but we don't know much about intergenic region. As a result, 98% they are non-coding region, and try to understand why they are there. Okay, coming back to the genes. Now, when you come back to the genes, again, we can find two regions. This is a one region in a white color. Oh, okay. Just a different color because okay, here another region. And maybe here another region. Like this. And if I take this gene, yes, there could be a region like this. 1, 2, maybe 3, 4, like this. Now, what does it mean? Gene has two different regions. A gene has two different regions. Now, I hope you understand between genes, the region is called intergenic region, which is non-coding. And now we are coming to the genes. Again, two regions. This is kind of these three represent one type of region, and these three represent one type of region.